Well, first off, you're going to hear from Gustav Karlberg. It's going to be a very excellent presentation. He is presently serving as Vice President and Life Cycle Leader at Roche, is a seasoned professional with more than 15 years experience in the omics field. He has a diverse background, and he has been at the forefront of driving advancements in human health through cutting-edge omics solutions. And so he's going to teach us everything we need to know about omics, and I'm very excited to have him here. So take it away, Gustav.
Okay, thank you so much. I really appreciate the opportunity, and thanks for the invite to participate here. So
there you go. Perfect.
Okay, perfect. Yep. Thank you very much. So thank you again for the organizers and for the invite. I really appreciate the opportunity to speak to you. I had the opportunity to speak at I can't remember if it was called Bridging the Gap at the time, but a couple of years ago I was also able to attend one of these events. So I'm really excited to be able to come back and speak a little bit more about genomics and omics in general, and all the things that we are doing around that and with that. And also my talk will touch on the interface between how all this genomics data is being used in healthcare and what we can do with AI and how we're utilizing that to draw insights and conclusions. Before I start, I just wanted to give you a quick overview of who Roche is. It's a Swiss pharma biotech company, multinational global company, really a big one. A couple of statistics that I think are pretty mind-blowing in a sense. 14.2 million patients have been treated with Roche medicines. We're very focused on the oncology space and other and some of those really difficult diseases, and I'll speak a little bit more about that. We're also a very big provider in the diagnostic space. So to date, 29 billion tests have been conducted with Roche diagnostic products. That to me is pretty mind-blowing. Just the breadth and width that we have of our technologies and the reach across the globe. But today I'm going to talk a little bit about genomics, and I want to start just with the science of DNA sequencing and speak a little bit to how does it work and why is it interesting and why is it important for us. And by starting by that, just want to introduce us to the genome itself and just give a perspective a little bit of just a challenge that we're facing when we're trying to understand the underlying molecules that is sort of the script for life, if you wish. It is very complex, the human genome. We have about 3 billion base pairs. And if you actually were to stretch out the DNA in one single cell, it's about 1.8 meters long. So you can imagine, knowing how small a cell is, how intricate this molecule has to be and how it's really bundled up in order to fit in there. Obviously, the human body has a lot of cells. And if we were to actually take all of that DNA that we have in us, as you can see here, we can go around the world quite a few times. So it's a really big molecule, long molecule, but a very, very interesting one because it allows us to understand a lot about what's happening in the biology and what's happening with us. So going in a little bit deeper, the DNA molecule really consists of 4 different bases. It's the adenine, thymine, guanine, and cytosine. And these bases are what's the way that we write sort of the book of life, if you wish. If you compare that to how we write things in our regular English language, or Swedish for that matter, we have 26 letters. Here, we're trying to make all these combinations out of just 4. And that has some challenges, but it also is a really nice way for us to understand the basics and the background of our code, of our DNA code. And this is actually what we're using in order to understand it a little bit more. But when we talk about biology, DNA is sort of, you should think of it as the blueprint of what we're trying to say, or the recipe, if you wish, if you want to put it into a recipe book. So it's the thing that describes what do we need to do, or what do we want this organism to look like. So in biology, we also talk about this sort of continuation from DNA and how that gets translated into RNA, and how RNA is really the instructions that we give, the recipe, and how we sort of say, and what are you going to build? from there, we go to amino acids and ultimately proteins, and this becomes sort of the building blocks of an organism, whether that is a human or whether that is something else. And this is the central dogma of biology, and really the key focus for all omics technologies is how do we understand this? How can we understand this process? What does it mean for us as an organism? What does it mean in spaces when it goes well? What does that lead to? And what is the variation within this? And I'll speak a little bit more to that today, because there is quite a lot of variation between human beings. I mean, we can see that when we look at each other. We see that we look different, but also on a molecular level, we have quite a lot of diversity in our genome, and that's important to understand. And it's also very important for us to understand the basics of what is sort of the quote unquote, I guess, normal way of DNA, and what happens when something goes wrong? What happens when we get a disease such as a cancer or something similar, where the genome might have had a change? And can we use that to understand what's happening in that particular disease? So I have a very, very, very simplified example here, where we just look at why is it important to be able to understand this, and why is it also to some extent important to understand the underlying difference in this. And if we look at the first one here, just saying you take one log, you take 1 ton of concrete, you take construction workers and you build a house. And that's what that's what the DNA is trying to say, very simplified. But what happens when something like this goes wrong? Well, it can go wrong in multiple ways, right? There can be an insertion of new letters where the instruction just becomes skewed, and therefore we don't end up with the right thing. Well, if we know what's happening, if we know what has changed, we have an opportunity to potentially treat that. And in some cases, these might be unique things. So in this very simplified, sort of trying to be a little bit funny example, this can lead to a particular disease, and we can change, we can do something, we can treat that, we can figure out what's happening. But what happens if there's a change that actually leads to a disease such as cancer? Cancer is a very complex disease, and this is where DNA really has a strong role to play, because different changes can lead to the outcome of cancer. But how do you know how to treat that the most effective way, and what do you want to do in order to really be able to address it? That requires us to have a molecular understanding of cancer. And this is where sequencing has really made a huge entrance into the space of medical treatment and understanding what can be done. Now, it's not just in cancer. I've spent a lot of time of my career also working in reproductive health and looking at being able to do things like non-invasive prenatal testing, which has now become standard. But sequencing really has this tremendous opportunity to understand at the molecular level, the starting level, what has potentially gone wrong here. And it can allow us to get a fuller insight to this. So how does sequencing work in more detail? I'm going to give you a very quick overview of it and try to cover a little bit of insights just to what is it that we do? How do we go about actually understanding this code of life and what is happening within a particular molecule? So this all starts with, it's really down to 3 steps in a simplified way, starting with preparing the sample, which means extracting the DNA, creating these readable pieces that we can then have introduced into our sequencing technology. I'm not going to go into a lot of detail about how this works, but there's a molecular preparation that happens where we actually take a sample, and we can either, in some cases, the DNA is already in small fragments. Otherwise, we can fragment the DNA, and we can put on these adapters that allow us then to use the sequencing technology that we want to use. And then we go to sequence it, and I'll show you a little bit about how that happens And then ultimately, becomes a really big puzzle. And if you think about those 3 billion base pairs that I talked about before that the human genome is consistent of, now we have very small fragments, maybe 200 base pair longs or 150 base pair long, or a little, in some cases, a little bit longer. But you still have to put all of those back together in order to be able to analyze the data and understand what is actually happening here in this particular individual. And this is where I think, data processing, algorithms, AI to a large extent have allowed us to open up ways of doing this much more effectively. And it's a very big area for us when we start talking about these molecular technologies. So in order to understand this a little bit more, you have to start thinking about where does the sample come from? That's really the first thing that people who come into sequencing and want to do an experiment have to think about is what kind of sample am I going to look at? Am I looking at something like a tissue culture or something where we have gone in and done a biopsy on a particular tumor and starting from that material? That will have its own unique challenges with how I prepare that sample. In some cases now, we can also use the blood to look at DNA fragments. We're talking about things like circulating tumor DNA. So in the cases where you develop a cancer, your cancer will actually drop DNA or shed DNA into the bloodstream. And we have an ability with the sequencing technology, because of the massive amount of data that we can capture with this, we have an ability to actually detect these things and be able to analyze it, understand what's happening at a molecular level without having to go in to do a biopsy, which is obviously very invasive. So this is an area in oncology testing that is really growing fast and has a lot of interest around the whole diagnostics field because of the simplicity of drawing blood compared to having to do a biopsy. Now, there's also obviously a lot of other things we can do with DNA. DNA is the founding molecule of every organism. It's not just the human body. So if we want to look at things like bacteria and other things, we can use the DNA sequencer to do that. At the end of the day, we're really trying to get into this complex matrix, break down the cells in some cases, and get this DNA out of there to then create a molecule that we can put into our sequencer. And that's what the whole sample preparation comes from. But the starting place for a sample is really important depending on what you want to achieve and what you want to do with this particular technology, or in this particular case, with the patient maybe, or research that you're doing. So I'm going to give you a little bit of an overview of then what happens in the sequencer. And if anyone participated in the presentation I gave a couple of years ago, I was working for Illumina at the time. I had a longer review of this. I'm not going to go into quite that detail today because we have a little bit of shorter time, but Illumina sequencing really, and now there's many others who also offer this type of sequencing, is based on introducing a base for every cycle. And then you introduce one base, it is capped, so you can only put one base on at a time. You use a laser to shine a light on the fluorescent label of that particular DNA base, and you can then detect the signal. So you get a pattern like this that's shown on the slide here in the flow cell, where you see a different light coming on depending on which base it is. And from there, you can identify and kind of read the bases molecularly and say, this was a T, this was a G, this was an A, and so on. And you do this cycle by cycle up until about 150 cycles. And that's the fragment that you read. Now, there's other sequencing technologies available too that does this, the same kind of thing, but in a different approach. And I'm going to give you a little bit of an insight to that. This is a nanopore technology, which really uses unique electrical signals to detect which base is being read. And it's doing that by using a nanopore, which is a biological construct that exists in the body as well. And what happens when you have a nanopore and you have these two different currencies on a different side of a membrane, you have this ion flow that you can see here on the left. And that flow would be consistent if the nanopore is open, so to speak, there's nothing in it. But what we do in this technology is that we introduce the molecule, and because there is now a molecule that blocks the nanopore, now the ion flow will change, and we can read that change and then identify which base it is. So every single DNA base, in this case, will have a different signal, a different kind of voltage, a different way of changing the voltage through the nanopore. And that can be detected. And that's really the basis of how you distinguish which base you're looking at. So two different ways of doing sequencing, ultimately with the same kind of output. You're reading the DNA and you're understanding which molecules are you seeing, and then you can start making sense of all that data. And I've talked a lot about this ability of creating a lot of data, and I wanted to just kind of show it to you on the next slide here. The way that most of these applications, when people are trying to understand the human genome, you want to sequence to a level where you cover this genome multiple times. So you remember we talked about the human genome being 3 billion base pair long. Well, for many of these applications, or to be able to understand what's unique about a particular individual, that 3 billion genome or billion bases reference genome has to be covered multiple times so that we can statistically determine if this change or this variant is correct. So that's the first thing we do. We sequence this genome to about 30 to 100x coverage. So if we do the simple math, right, 3 billion times 100 times, that's a lot of DNA bases and a lot of data that we are producing for each and every run. Now, the sequencing technology has really made a tremendous evolution and gone from being able to do very small amounts of data. When I started in next generation sequencing, the sequencer actually did 3 gigabases of data. That is a long, long time ago now in the sequencing world. Now we can actually do up to about 96 whole human genomes covered 30 times in one single run. So the evolution of data generation here has gone really, really fast. From all these little short reads, what we do is we then align this back to create this 3 billion base pair genome. And like I said, we want to be able to stack reads on top of each other so that we can truly see if this is a true variant, or in some cases where the sequencer might have made an error, we can correct those errors. And then we finally get to the genome, and I hope you can see this on the screen, where you have these individual variants. And just for a reference, when you compare any one of us, we have about 0.1% of our genome that is unique to ourselves. Now, that doesn't sound like a lot when you talk about 0.1%, but if you put it in context of 3 billion bases, it's quite a lot of variance. And if my math is serving me now, I think that will end us up somewhere around 3 million unique variants for our genome. So there's a lot of data here to process. Now you can imagine the challenge and the complexity when you're trying to then also maybe determine in a case where someone has a disease, what is unique and what is different and what really makes a difference for that particular disease. And this is where sequencing and all this data processing and AI really has starting to converge really rapidly. And we've been using, first of all, we use a lot of different machine learning in order to do this whole process as efficient as possible. But then also as we get to the next stages of trying to understand this, what does it mean? It really becomes a powerful tool for us in this field. So when you think about sequencing, I wanted to bring up a couple of really interesting applications that are emerging in this field. And for myself, some of the reasons why I was interested in working at a company like Roche is also we have such a breadth of things that we do internally. So we're very well positioned to do a lot of these things. But I want to talk a little bit about just how do people use this information, and what are the thoughts in the field of how sequencing is going to change the way we do things. If we start in the world of pharmaceuticals and drug discovery clinical trials, this technology is being used now to do a lot of biomarker discovery. So imagine you have a big group of people, you have a set of people who have a certain disease, you have a set of people who are healthy. Well, now you can use this technology, identify those variants, and compare those different groups and see, are there molecular drivers to this disease that I can identify? And can that give me an insight then to how I should think about treatment? The other part we talk a lot about is stratification. And stratification is being able to build groups, I guess, of people who have a similar molecular composition. So a lot of times when in the past, when we used to do this, we used to look at people and say, kind of who looks similar. Well, now we don't have to do that. We can actually look at the DNA and say, is there a molecular similarity between different groups? Because in some cases, medicine actually doesn't work well for certain people. Certain molecular configurations are just not well suited for certain treatments. And stratification of groups that way allows us to help doing that a little bit more. Also, monitoring is a very interesting evolving field that is happening both in pharmaceutical clinical trials, which means, can I see what happens in this particular patient after I treat them with a certain drug? Does that change their composition on a molecular level? Does it start changing what happens with the disease? And if we go to the next row here and talk about oncology, monitoring has become a really interesting field for reoccurrence monitoring. So imagine that you have the unfortunate event of going through a cancer, but you get treated. Now we know that a lot of times, unfortunately, cancer is one of these diseases that will reoccur. And we can use this molecular technology to monitor that and be able to pick that up early and then be able to treat it. That is a really powerful tool in order to improve the health of people and have better outcomes. One of the first areas, though, that emerged for sequencing as a real tool was this companion diagnostics therapy selection. And I think this was really one of the first time also we showcased the capabilities of personalized medicine and really at a molecular level what that means. So this is the case where today, if you are diagnosed with cancer, we can actually do a genomic test on that particular cancer. And rather than the previous standard of care, which was to say, your cancer is situated in this particular tissue, whether that would be your kidney or it would be your liver or something like that. That's actually how we classified cancer. At this point now, we can do a molecular test, determine what the molecular composition of that cancer is, what's driving the emergence of this cancer, and then treat that molecular signal. So maybe that means that a drug that maybe was developed for a kidney cancer can be used for someone who has a totally different type of cancer, like a lung cancer or similar. But because of the molecular composition of the tumor, we know it's going to be effective or likely to be effective on that particular cancer because it's the same molecular composition as the original cancer where the drug was developed. So this is a really powerful tool to drive personalized medicine. Now, for the last one here, early detection and diagnosis, this is really where we see the huge opportunity of sequencing and being able to detect cancers very, very early and treat them at an early stage. This is something that a lot of people are working on just from a simple blood sample, being able to pull this out and identify people who are going through or developing a cancer at a much earlier stage, where also we know that treatment is much more likely to lead to a positive outcome. So I'm going to go through a little bit quicker here because I know I'm running out of time. But just oncology is a very complex disease. I've talked a lot about the genomics of this. We do have all the other aspects of this too, where sequencing can be used. And I'd love to come back and talk more about how we use it for transcriptomic, proteomics, etc. But really maybe leave you with this message that in order for us to get a deep understanding of what happens in biology and the complexity of biology, we need to look at lots of different aspects of this, and we need to be able to interpret that. I'm going to skip over this pretty quickly because I know I'm running out of time, and I'm going to be told that I've talked too much. But from a perspective of AI and how we are utilizing this data, it is really about that ability of combining data from multiple different sources and then being able to use these algorithms, have AI help us drive this. So we do combinations of omics data, time series, and imaging in order to be able to draw the right conclusions from this. So where does this leave us? I do want to leave with this because I feel like when we talk about AI, there's always this question about, well, what does that mean? Is it going to mean that we're going to remove people? We don't need people to be involved in this. I really don't personally believe that is the case. I think AI is an amazing tool in order for us to interpret results, manage diseases, being able to take all this massive amounts of data, help us distill it down, help with drawing the right conclusions. And the reason we need that is I use this as an example here on the left. This is just a graph showing you as sequencing costs have gone down in the gray here, how much publications have sort of grown over time. And why is that important? Well, to me, I think it's putting a real challenge on the healthcare problem here. How do you, as a physician or someone practicing medicine, keep up with all the new information that's coming out? That's where I think really bringing AI together and having it as part of how do we generate the right data, but then also how do we interpret that and how do we actually help make sense of all the information that's coming at us. And thank you again. I'm going to stop there. I apologize if I ran a little bit over, but I appreciate the opportunity.
This was wonderful, Gustav. I really like how you broke it down and seeing the various sources of data and how you're combining it. I think you did a very compelling case for how AI can help us make sense of the data and use it not only for treating, but early diagnosis and making sure the patient gets the right medicine. So thank you for that. I'm also looking forward to when you get to talk again at the panel discussion and have all of you interact. So wonderful, wonderful presentation. Thank you. So